Hey guys, welcome to part two of Young-Splaining photosynthesis. So, just as a recap, I'm going to quickly go over part one, which talks about the linear and the cyclic, but I'm going to do it with the linear example. So, what you see here is a cross section of a thylakoid. And so you have your thylakoid membrane, and this is your thylakoid space. And of course, outside of the thylakoid, we have the stroma. So this is kind of the big picture, guys. So this is going to be photosystem 2, and this is photosystem 1. So remember that 680 nanometer light is going to be captured by these pigment molecules, and that energy is going to be passed to that chlorophyll, that P680. The electron gets excited and it hops up to the primary electron acceptor, making P680 P680 plus, which is the strongest known biological oxidizer. And water is going to be split during this process. When water is split, the oxygen is released as a byproduct because we don't want that. The hydrogens are going to be stripped of their electrons, and those electrons are going to go into that chlorophyll A to reduce P680 plus back to P680. Those hydrogens become hydrogen ions, or what we call protons, because at this point they only contain a proton. And those electrons are going to be passed down in an electron complex, cytochrome complex. Now, this drop in energy, this electron being passed through these cytochromes, is going to actually drive that proton force. These protons are going to cross the thylakoid membrane. As they cross the thylakoid membrane, they're going to be instrumental in the actions of ATP synthase. ATP synthase is the enzyme that drives the formation of ATP from ADP and inorganic phosphate. Now, also what's going to be happening is photosystem 1 is absorbing at 700 nanometer wavelength light, and P700 is getting excited and it's jumping up its electron and becoming P700 plus. And so I have the negative electron here being pulled to this photosystem and this reaction center to reduce P700 plus. That electron is going to be passed to another electron transport, this time the ferredoxin. Ferredoxin is going to pass those electrons through NADP reductase, which is going to also use NADP plus and pull some of those protons from the stroma to synthesize NADPH. If you notice, both ATP and NADPH are being synthesized on the stroma side, even though their synthesis is driven by the reactions of the thylakoid happening in the thylakoid membrane. The actual products are being produced on the side of the stroma so that they can be used in the Calvin cycle. Now, let's talk about the Calvin cycle. The Calvin cycle is, of course, light independent. It's light independent because the energy source for the Calvin cycle is the ATP that was created during the light dependent. So the Calvin cycle's job is to take inorganic carbon in the form of carbon dioxide and reduce that to organic carbon in the form of a sugar. Now, I know in lower level science classes we say that sugar is glucose. Not exactly true. The immediate product is glyceraldehyde 3-phosphate, which it takes 2 glyceraldehyde 3-phosphates to make a glucose, which is a C6 compound. It has 6 carbons. Now, we call normal plants undergo what's known as a C3 process because it takes carbon dioxide and incorporates it to make a 3-carbon compound. 3 carbon dioxides enter one glyceraldehyde 3-phosphate leaves, and then the remaining compounds are used to regenerate the carbon dioxide acceptor. So let's take a closer look at the Calvin cycle. So the Calvin cycle starts with RUBP, which is ribulose bisphosphate. RUBP is what we call the carbon dioxide acceptor. Now, the way that RUBP is going to be able to actually accept the carbon dioxide, that reaction is going to be catalyzed by the enzyme ribisco. Ribisco has an affinity for carbon dioxide when it is abundant in the cell. When the levels of carbon dioxide decrease, ribisco is no longer able to catalyze the fixation of carbon dioxide. So the first step is carbon fixation. If you notice, we're going to do 3. This is showing 3 at a time, entering 1 per cycle. So it takes 3 cycles for this to happen. What we're going to just go ahead and combine it all together. Now, if you look at RUBP, it is a 5, 1, 2, 3, 4, 5 carbon molecule. Carbon dioxide contains 1 carbon. So we're essentially taking this 5 carbon molecule, attaching 1 carbon, making it into a 6 carbon molecule. Another thing I want you to notice about RUBP is that there is a phosphate on both ends. Okay, that's why it's ribulose bisphosphate. Phosphates on both ends. There's 2 phosphate groups. So now we have, in carbon fixation, we take this 5 carbon compound, add a 1 carbon compound to make a 6 carbon compound. This 6 carbon compound is going to be broken down into, instead of having 3 6 carbon compounds, I'm going to end up with 6 3 carbon compounds. Still 18 carbons, okay? Keep up with your carbons. We're counting carbons. But instead of having a compound that has a phosphate on both ends, it now only has a phosphate on one end. And this compound is called 3-phosphoglycerate. Now, through the process of carbon fixation, what has to happen is I'm going to use, at the expense of 6 ATP, because I need 6 more phosphates so that I can phosphorylate the other end of that compound. So I want to make 3-phosphoglycerate become 1,3-bisphosphoglycerate. And these numbers 3 and 1,3 are telling you which carbon is located on. In this case, it's on the number 3 carbon. Here you have it on both the number 1 and the number 3 carbon. So 1,3-bisphosphoglycerate, 3 carbon compound, 2 phosphates, 1 on the number 1, 1 on the number 3. We're going to have 6 of those. This 1,3-bisphosphoglycerate needs to be reduced. So hydrogen needs to come in. It needs to bring in those electrons that come with it. And since there's 6 of these compounds, it's going to take 6 NADPH to reduce 1,3-bisphosphoglycerate. So again, this is the ATP and the NADPH that came from the first reaction, the light reactions. Now, this NADPH is going to reduce 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate. So now I have 6 glyceraldehyde 3-phosphates, but here's the thing, only 1 is going to come out. After those 3 turns, I'm going to generate a total of 6 of these compounds, but only 1 drops out because I started off with 15 carbons. I have to end this cycle with 15 carbons. How many carbons did I put into it? 3, with the 3 carbon dioxides. So this 1 3-carbon compound comes out. It can be used to make glucose and other organic compounds. After this output of 1 glyceraldehyde 3-phosphate has occurred, I have to regenerate my carbon dioxide acceptor. So my carbon dioxide acceptor, so these 5 glyceraldehyde 3-phosphates. So I have 5 3 carbon compounds. That's 15 carbons. How many carbons did I start off with? 15 carbons. But what am I missing? I'm missing the phosphates. So it's going to take 3 more ATP to phosphorylate these compounds. And I'm going to change 5 3 carbon compounds into 3 5 carbon compounds again. I do that through the regeneration of RUBP. And when this happens, I have to use 3 more ATP. So to get 1 glyceraldehyde 3-phosphate, I need 9 ATP and 6 NADPH. So in order to make 1 molecule of glucose, it's going to take 18 ATP and 12 NADPH. Because remember, this molecule is only half of a glucose molecule. So in the Calvin cycle, carbon enters as carbon dioxide and leaves a sugar named glyceraldehyde 3-phosphate. For 1 glyceraldehyde 3-phosphate, this cycle must take place 3 times. That's to fix 3 molecules of carbon dioxide. There's 3 phases: carbon fixation, reduction, and regeneration of ribulose bisphosphate, or RUBP. Now, what happens when it's hot or arid and plants are becoming dehydrated? So sometimes they have to do what's called a trade-off, and they may end up doing what's known as photorespiration. Now, in photorespiration is a wasteful process. In photorespiration, the plant is going to close its stomata. It's going to close its stoma so that it does not lose water through transpiration. But in closing the stoma, I'm causing a buildup of oxygen and I'm not allowing carbon dioxide in. So I'm keeping oxygen in and keeping carbon dioxide out. So this leads to what's known as photorespiration. Now, photorespiration is not good for the plants. So what some plants have done is, okay, so what is photorespiration? Well, in photorespiration, instead of rubisco catalyzing the acceptance of carbon dioxide, oxygen is actually fixed. And so we don't end up with a C3 compound, we end up with a C2 compound. And that's a wasteful process because when this happens, at least 50% of the carbon dioxide is used up. Like it's, so it's a wasteful process, to say the least. Now, what some plants have done is they have evolved a different way to deal with this and to avoid photorespiration. And some of those plants are what we call C4 plants. So what they do is they create an organic acid. C4 plants make a organic acid to minimize the cost of photorespiration. C4 plants use a different enzyme, and this enzyme actually has a higher affinity for carbon dioxide. So where rubisco can only bind carbon dioxide at high levels, this enzyme can bind carbon dioxide at low levels. And so C4 plants use a spatial separation of the steps. Remember those mesophyll cells and the bundle sheath cells from before? The bundle sheath cells are closer to the xylem and the phloem. So what happens with the mesophyll cells is the carbon dioxide is incorporated into an organic acid in the mesophyll cells. So instead of making the sugar, it makes an organic acid. And then it moves that acid from the mesophyll cells to the bundle sheath cells where they release carbon dioxide. And that carbon dioxide is then used in the Calvin cycle, and the sugar is passed on to the phloem. So that aids in reducing the effects of photorespiration. The enzyme is PEP carboxylase, PEP carboxylase. And PEP carboxylase, again, has a higher affinity for carbon dioxide. So it can actually bind that carbon dioxide in the lower concentrations than Rubisco can. Now, another way that plants deal with this, so C4 plants use a spatial arrangement. They use the mesophyll cells and the bundle sheath cells. CAM plants, actually, which is crustacean acid metabolism, CAM, crustacean acid metabolism. These plants, plants like succulents, what they do is they just open their stomata at night. So instead of opening the stomata in the daytime, it opens the stomata at night to allow carbon dioxide in, and it incorporates that carbon dioxide at night into an organic acid. In the daytime, when the stomata or the stoma is closed, that's when it actually does the Calvin cycle by releasing the carbon dioxide from the organic acids. So there's two ways plants have evolved to deal with reducing photorespiration. Remember, most plants are C3 plants where they're going to make a 3-carbon compound. C4 plants are going to make a 4-carbon organic acid, and they do that by using PEP carboxylase or PEP carboxylase in the mesophyll cells to fix the carbon dioxide into a 4-carbon organic acid first, and then move it from the mesophyll to the bundle sheath, where the acids can release the carbon dioxide for the Calvin cycle. Or you have plants that open their stomata at night, so that's a temporal separation. So instead of opening in the daytime, the stomata opens at night to take in the carbon dioxide. It's cooler at night. You lose less water at night. So again, that is photosynthesis in a nutshell. I hope that helped.